Phospholipids, you can see in the top picture, the top right picture, there they have a hydro, hydrophobic, hydrophilic head, which means that it's water loving, and that's why it's facing the outside, which is towards the water. And then they have a hydrophobic tail, and hydrophobic means water fearing. So that's why it's facing the inside. It's kind of like escaping the water. And then it's also amophilic, so that means it has one surface consisting of hydrophilic amino acids and the opposite surface consisting of hydrophobic ones, which is kind of what I just went over. And then the bottom right picture, it's a bigger picture of just one of the phospholipids, and then there's two like in the actual cell membrane. And then kind of similar to this, um, the structure of a cell membrane, it's modeled by a fluid mosaic model. So that's the picture on the top right. That's called a fluid mosaic model. And as I said, it consists of phospholipids, cholesterol, which you can see are the yellow dots in the bottom picture, proteins, which are the big blue things, and proteins can act as channels, letting molecules pass through. And I think we go over that today. And then also carbohydrates, which are the little tails attached to the proteins. And as I said earlier, it has a bilayer structure. So there's two phospholipids, and that's what gives its fluidity. And that's why it's called a fluid mosaic model.
So there are many membrane proteins on the phospholipid bilayer, and these proteins are really crucial to the function of the membrane. So first are integral proteins, which penetrate the inside of the bilayer, and they are also called transmembrane proteins because they pass through the whole membrane, kind of like this. And peripheral proteins are bound to only the surface of the bilayer, which is why they're on the peripheral. And protein channels are another type of protein on the membrane, and these allow for the passage of hydrophilic or polar materials because hydrophilic and polar materials don't easily pass through the membrane, and these proteins allow them to pass through it really easily. And next are glycoproteins, which are proteins with carbohydrates bonded on the surface over here. And for example, you could have AB, and O markers, which are characteristic of your blood. So these proteins are crucial for helping your cells recognize that they are your own cells made by your own body. And finally, extracellular matrix, which consists of proteins bonded to fibers to help support and for structure. So these are all on the outside of the cell, and they're usually connected with the proteins that are usually transmembrane proteins, and they are useful for keeping like a layer of protection for the cell. Okay, so for selective permeability, the cell is able to take up some small molecules and ions and exclude others. So nonpolar molecules are hydrophobic, like the membrane, so they cross the bilayer easily, while polar molecules cross very slowly. And hydrophilic substances move with the help of transport proteins because they do not cross the bilayer very easily. And aquaporins help our channel proteins for moving water because water is polar, and carrier proteins change shape to shuttle substances across the membrane, which we described earlier. And finally, transport proteins are specific for the substance being translocated. So transport proteins have a specific shape that allows it to transport only a certain type of substance. Okay, so finally, I'm going to go over passive and active transport. Passive transport is basically diffusion, which is the movement of solids from high to low concentration. So it's like kind of down the concentration gradient. And facilitated diffusion is where protein channels or other types of channels allow solutes to move down the gradient, even though the solutes can't pass through the membrane like naturally. And over here, you can see the different types of passive transport. So there's carrier mediated, channel mediated for facilitated diffusion, and then free for like osmosis for simple diffusion. And active transport is basically moving things against the concentration gradient, so from low to high concentration. And primary is creating an electrochemical gradient using these types of proteins. There's uniport and co-transport, and we'll cover these later in a proteins proteins chapter, I guess. And then next is secondary, which is where a gradient is used to transport molecules across a membrane. So basically, after these primary transporters like move all of the proteins against, move all the solutes against the gradient, the secondary transport will just allow the proteins to move back down the concentration gradient. And finally, exocytosis and endocytosis are also active transport, but these are involved using the actual cell to create a vesicle around the substance that the cell wants to like eat. And then that allows it to just take in the substance without using proteins or other types of channels. And endocytosis is like the process of eating it, while exocytosis is releasing substances. Okay, since Grace, you're the only one here, we're not going to do like a hoop. Yeah, for homework, we're going to do a review worksheet and an activity in Google Classroom. And yeah, thank you for coming.
Do you have any questions? Like, it can be about this topic or like just the class in general, or like any like. Are you taking biology in school? No? Okay. I was asking that because like if you were, we could also like help you with like homework or anything you want. But if you're not, then I guess you don't have any homework. Thank you for coming.